Welcome to the deep dive. Every single species out there, whether it's tiny like a microbe or huge like a blue whale, it has a boundary, right? A geographic limit. It stops somewhere. And that's not just like a neat biological fact. It's actually a really critical research area, especially now with climate change happening so fast. Understanding where a species can live and maybe more importantly, where it can't. That's vital for conservation efforts, wouldn't you say?
Oh, absolutely. And that's why this deep dive is focusing on a really significant paper. It's from 2025 by Tom Radomski called The Ecology of Geographic Range Limits.
Mm.
For a long time, this field felt a bit stuck. Researchers acknowledged we had loads of theories, competition, climate, dispersal, but actually proving which one was the key factor in nature. That was tough. There was this feeling of, well, rich theory, but maybe not enough solid empirical work to back it all up clearly.
Okay, so let's unpack that. Radomski's paper is arguing for a shift then, a new way of looking at this.
Exactly. He's suggesting we need to move away from testing, a dozen different ideas all at once for a single species. Instead, he proposes what he calls a first principles approach. The idea is to bring some clarity by boiling down all those complex models into just a handful of core, non-overlapping processes.
Right, simplify things, get back to basics.
Pretty much. Radomski's framework suggests that basically any range limit comes down to how a species interacts with maybe just 5 key things. Its basic habitat needs, any helpful interactions with other species, harmful interactions, hybridization, and its ability to disperse or move.
5 categories. Okay, that does sound more manageable than, endless possibilities.
If we test those systematically, the argument goes, our understanding should get a lot better, faster.
Okay, so if those are the big 5, where does Radomski say we have to begin? What's the absolute starting point, the real first principle?
He calls it the Xenopoetic Niche Limits Hypothesis, SNLH for short.
Xenopoetic niche. Okay, bit of jargon there.
Right.
What does that mean for us?
It's actually a really simple, elegant idea. The xenopoetic niche is just the range of environmental conditions where a species can survive and reproduce based only on its tolerance to non-consumable habitat factors.
Non-consumable, meaning things it doesn't eat or use up.
Exactly. Think temperature, rainfall, salinity, maybe soil pH, or even physical structure like forest canopy cover. Critically, we're deliberately leaving out other living things for now. No competitors, no predators, no food sources in this initial step.
Ah, okay, so it's the absolute baseline, like the fundamental physical limits of the organism itself.
Precisely. Every species has limits. If it gets too hot, too cold, too salty, it simply can't persist, regardless of anything else going on.
That makes a lot of sense as the foundation. Do we have some classic examples where this xenopoetic niche is clearly the main driver of a range limit?
Oh, definitely. Cold tolerance is a great one. Think about the saguaro cactus, Carnegie gigantea, in the American Southwest. Its range stops pretty sharply to the east and north, and that limit matches up almost perfectly with how long freezing temperatures last. If it stays below freezing for too many hours consecutively, the cactus tissues get damaged beyond repair.
So it doesn't matter if the soil is good or there are no competing plants. The frost itself sets the boundary.
Exactly. And you see the same sort of thing with the timberline on mountains or in Arctic regions.
Right, where the trees just stop growing higher up or further north.
Yeah. That line is largely determined by temperature during the growing season, often related to things like root damage from cold soils. The abiotic environment is the hard stop. And the beauty of the SNLH is that it forces us to really test this baseline environmental limit first, before jumping to more complex explanations like competition.
Okay, so how do you test this? You can't just plant a saguaro somewhere cold and wait 50 years, right? How does it work in practice?
Well, typically you start with modeling. One common approach uses correlative ecological niche models, or EMMs. These take data on where the species is currently found, layer in environmental data like climate maps, and then predict other areas with similar conditions where it should be able to live.
But isn't there a risk of just modeling what you already know? If you only use places it currently lives, aren't you just finding correlations?
That's the big caveat, yes. There is a real risk of circular reasoning, or maybe over-interpreting those correlations, which is why the, let's call it the gold standard, is using mechanistic ENMs.
Mechanistic. How are they different?
Instead of relying only on where the species is, these models use independently measured data about the organism itself, its physiology, its life cycle needs, its actual tolerances. Things measured in a lab or field study. They try to predict the distribution from the ground up based on biological mechanisms, not just correlations.
Okay, that sounds much more powerful. Can you give an example where a mechanistic model really nailed it?
Yeah, there's a great study on a butterfly, Atelopedes campestris, in the US. Researchers built a mechanistic model based on just two key physiological traits they measured. First, how long the overwintering larvae could actually survive freezing temperatures. And second, how many degree days, like heat units, the summer generation needed to successfully reproduce. They plugged these biological thresholds into climate data, ran the model, and voila, it accurately predicted the butterfly's known northern range limit.
Wow. So the model, based on its cold tolerance and heat needs, perfectly matched where the butterfly actually stops.
Exactly. It strongly suggested that, in this case, the xenopoietic niche, specifically winter cold, was indeed the limiting factor. No
Hmm.
need to invoke competitors or anything else.
That's really compelling. But as you mentioned, Radomski argues the real insight comes when this doesn't work, right? When the SNLH fails.
Precisely. The power of using this as the first step is that how it fails points you directly to the next likely cause. If the model gets it wrong, if it can't separate occupied sites from unoccupied sites, well, then, something else is going on.
And there are two main ways it can fail.
Yes. Let's look at failure mode 1 first. Model under prediction.
Okay, under prediction. So the model says, nope, too harsh here. This species shouldn't survive. But surprise, it's actually living there.
Right. The observed range is bigger than the xenopoietic niche predicted by the model. The species is found in places that seem, based on its basic tolerances, unsuitable.
So what causes that? How can it live where it shouldn't?
Well, one common reason is what we call sink populations. These are patches of habitat where the species can't actually sustain itself long-term through reproduction alone. They only persist because individuals are constantly arriving from nearby, much better source habitats where reproduction is high. Constant immigration props them up.
Okay, so they're living on subsidies from elsewhere. What else?
Radomski highlights this really interesting and maybe slightly worrying concept called habitat debt.
Habitat debt. I like the sound of that. What does it mean?
Think of relic populations. These are individuals, often long-lived ones, like trees, that established when the environment was suitable. But then the environment changed. Maybe climate shifted, maybe a key resource disappeared faster than the organism's lifespan. So the old individuals are still there, physically persisting, but they aren't successfully reproducing anymore. The conditions are no longer right for new recruitment.
Wow. So they look like a healthy population, but they're essentially ghosts, living on borrowed time because the environmental rug was pulled out from under them years ago.
Exactly. It's a lag effect. They haven't died off yet, but their fate might already be sealed because the conditions needed for the next generation are gone. It's a huge challenge for conservation. You might think an area is fine based on current adults, but it's actually doomed.
That's a sobering thought. So sink populations, habitat debt. Any other reasons for underprediction?
Yes, positive interactions. Sometimes having a friend allows a species to push beyond its fundamental limits. Mutualisms are key here. There's a great example with Chinook brome, which is a type of grass, Bromus levipes. Researchers found that a specific mutualism with a soil fungus allowed this grass to expand its range significantly, we're talking thousands of square kilometers, into areas where the climate conditions alone should have prevented it from surviving. The fungus helped it tolerate the harsher conditions.
Okay, so failure mode 1,
Yeah.
the species being more widespread than predicted, points towards things like immigration maintaining sink populations, the lag effect of habitat debt, or positive interactions like mutualisms helping it out.
Correct. It relates back to Radomski's categories.
Yeah.
Maybe it's about habitat distribution dynamics or sink or positive biotic interactions.
Right. Now for the other side of the coin, failure mode 2, model over prediction.
This is when the model says, hey, this area looks great. Perfect climate, perfect conditions. The species should thrive here, but it's not there. The actual observed range is smaller or truncated compared to what the xenopoietic niche predicts.
So suitable habitat is left unoccupied. Why would that happen? This seems like where the other factors must come in.
Exactly. This failure mode points squarely towards those other categories in the framework. Antagonistic biotic interactions, like competition or predation, limitation by a consumable resource, limits imposed by hybridization, or simple dispersal limitation. Let's tackle the antagonists first.
Okay, so competition, predation, parasitism, disease. Radomski groups these together. Why?
Because fundamentally they all do the same thing. They shrink the set of conditions where a species can actually persist. Even if the climate is perfect, a superior competitor or a devastating predator can just exclude it.
Makes sense. When we think about competition setting range limits, what's a common pitfall in how researchers interpret patterns?
Oh, the classic trap is looking at where two species ranges meet along an environmental gradient and assuming competition is automatically the cause and maybe strongest right there. But Pilou pointed this out way back in the 70s. The pattern you see, how much the ranges overlap or how sharp the boundary is, can be heavily influenced just by how steep the environmental gradient is, independent of the actual biological interaction strength.
Wait, say that again. The steepness of the environment can trick you into thinking competition is stronger or weaker than it really is.
Yeah, exactly. Imagine two species, A and B, competing along a temperature gradient. If the temperature changes really slowly over a large area, a shallow gradient, the ranges might overlap quite a bit, even with strong competition. It looks like they coexist broadly. Now, picture the same two species with the exact same competitive interaction, but on a steep mountainside where the temperature plummets over a short distance, a steep gradient. Their geographic zones of dominance will be narrow, and the overlap tiny. It looks like sharp competitive exclusion.
Ah, I see. The map pattern looks completely different purely because of how quickly the environment changes. Even if the underlying biology, the competition itself, is identical in both scenarios.
Precisely. So you can't just look at the map, you have to consider the environmental context. It's a crucial point, often overlooked.
That really changes how you'd interpret range boundaries. Okay, what about predation and disease? How do they limit ranges, especially indirectly?
Predation often works through environmental gradients affecting the predator, not the prey. There's a good example with wolf's bane, the plant Arnica montana. Its lower elevation limit isn't set directly by temperature being bad for the plant, but because warmer temperatures down low allow slugs which eat the plant to be much more active and abundant. Go higher up, it gets colder, the slugs become less active, and the plant survives.
So the plant's range is limited by the slug's climate tolerance, essentially.
Exactly. The predator's niche shapes the prey's range edge. Disease can be similar, but often involves density dependence.
Density dependence, meaning it depends on how crowded the host population is.
Right. Many diseases need a certain host density to transmit effectively. Theoretical models predict that at the very edge of a species range, where populations are naturally less dense and patchier, the density might drop below the threshold needed for the disease to persist. This can create what's sometimes called a disease-free halo right at the range margin. The species can survive there partly because its main pathogen can't gain a foothold.
Like the hosts are too spread out for the disease to easily jump between them. That's fascinating.
It is. It shows how dynamic these interactions can be.
Okay, quick thoughts on the next two causes of overprediction: consumable resources and hybridization.
Consumable resource limitation is usually pretty straightforward, especially for specialists. If a species absolutely depends on a specific food source or a host plant or nesting site, its range simply cannot extend beyond the range of that critical resource.
The consumer follows the resource. Makes sense.
The classic example is an insect that only feeds on one type of plant. No plant, no insect, even if the climate is perfect. Hybridization limits are a bit different.
This is when two related species mate, but the offspring aren't viable or are less fit.
Yes. If the hybrids produced where the ranges meet have significantly reduced fitness or are sterile, it creates selection against interbreeding. This can maintain a sharp boundary between the two species, often called a parapatric range boundary.
Parapatric, meaning they meet but don't overlap much.
Right, they abut each other. And this often happens near range edges because individuals might have fewer choices for mates and end up hybridizing more readily than they would in the core of the range where partners of their own species are abundant.
Okay, that covers resources and hybrids.
Okay.
The last big reason for overprediction was dispersal limitation. Radomski emphasizes this term gets used in a few different ways. Can you clarify?
Yeah, it's important to distinguish. First, there's the simple failure to reach a suitable patch. Maybe it's an island, maybe it's a habitat fragment far away. This can be linked to alley effects, too. Sometimes you need a certain minimum number of individuals arriving together to successfully establish a new population. One seed might not be enough.
So just not getting there, or not getting there in sufficient numbers. What's the second meaning?
This one's crucial. Time-lagged dispersal. This is kind of the opposite of habitat debt. Here, suitable habitat is available. Maybe the climate has warmed up and opened new areas, but the species simply hasn't been able to move fast enough to colonize it yet.
Ah, so the species is lagging behind the expansion of its potential niche.
Exactly. This ties into something called Reed's paradox.
I've heard of that. It's about the puzzle of how trees, for instance, seem to migrate incredibly fast after the last ice age, much faster than we see trees spreading today, right?
Precisely. It highlights this potential mismatch between how fast environments can change and how fast species can actually track those changes through dispersal. Just because the door opens doesn't mean the species can walk through it immediately.
Okay, failure to arrive, lagging behind. What's the third type of dispersal limitation?
Current determined dispersal. This is when external physical forces actively shape the range by influencing movement patterns. Think persistent winds blowing seeds only in one direction, or strong ocean currents carrying larvae away from suitable coastlines, or maybe concentrating them in less ideal areas. The physical movement of propagals itself dictates the range edge.
Got it. So dispersal isn't just about ability, but also about timing and external forces. Now, before we wrap up with the research agenda, Radomski specifically cautions against focusing too much on one particular idea as a cause of range limits. What was that?
Right, the center-periphery hypothesis. This hypothesis predicts certain patterns you might see as you move from the core of a species' range towards its edge. Things like lower population density, maybe less genetic diversity, perhaps individuals in poorer condition.
And people often assume these patterns explain the limit.
Sometimes, yes, or they focused heavily on testing these predictions. But Radomski argues that while these patterns might exist, and the evidence is actually pretty inconsistent, except maybe for lower density, they don't tell you why the range stops there. There are potential consequences of being near a limit, not the cause of the limit itself.
So it's like observing that people near the edge of a desert are thirstier. That doesn't explain why the desert ends. It's just a correlation with being near the boundary condition, which is lack of water.
That's a perfect analogy. The center-periphery idea describes potential correlations, not the underlying mechanism. The lack of water, the lethal temperature, the competitor that actually sets the limit. We need to focus on the mechanism.
Okay, so given this first principles framework and the pitfalls to avoid, what's the research agenda Radomski proposes? How should the field move forward?
Well, two main things. First, we desperately need more rigorous empirical studies that are explicitly designed to test the SNLH first, and then, based on how it fails, systematically test the next plausible factor, really teasing apart that xenopoetic baseline from everything else.
Stop lumping all niche limits together. Be specific.
Exactly. Second, we need more focused work on the role of those biotic interactions, especially the antagonistic ones, and crucially, incorporating time and space dynamics. Understanding how things like competition or disease play out over time may be leading to unstable boundaries or pulsed range shifts.
And bringing this back to why we started climate change, this systematic approach feels incredibly important. If we want to predict how ranges will shift, we absolutely need to know why they are where they are now, right?
Absolutely. You can't predict the future if you don't understand the present mechanisms. The SNLH framework gives us a logical starting point and a roadmap. When it works, great. When it fails, it tells you which road to take next. Investigate sinks, habitat debt, mutualists, competitors, resources, dispersal. Of course, it's not always easy. Field ecology has real logistical constraints. Measuring lifetime fitness is hard, so researchers often rely on proxies like physical condition or local abundance. But the ideal path forward combines careful observation with those mechanistic models we talked about, and targeted field experiments to really nail down causation.
It provides a clear, testable structure for a complex problem. So here's a final provocative thought for you, drawing on this discussion. If that time-lag dispersal is a common phenomenon, and species often struggle to keep up as their suitable climate shifts, how many species that look perfectly healthy and widespread today might actually be sitting on an extinction debt? Not because their current habitat is bad, but because the suitable areas they needed to colonize years ago are places they just haven't reached yet, while their old range slowly becomes unsuitable behind them. Are they running out of time without us even realizing it? Thank you for joining us for this deep dive.